So the bottom incision, so the whole cuts here are to put in cameras and equipment, and then the suprapubic area here would be where they would pull the kidney out. That's usually about 3 inches. And so when I was getting these, they actually they do a few in women transvaginally.
Wow.
Really?
Oh my goodness.
That's a long
You
way
would have
to
to
go.
cut the vagina to pull it through, because you can't pull it through the oviduct, obviously. But I'm not sure. So that's one way it plays. Another is through the umbilicus. Okay. Or they may cut to the side. There's a single incision through the umbilicus. The other one was a larger incision that the surgeon could put his hand in and manipulate it, and they would bring the kidney out that same size. Or here they just have one cut, and then all the equipment, the cameras, and the surgical instruments go through there. So here's where they place it.
Oh, they're going lower.
So otherwise,
On the,
it would be much more likely to shift and drop. So they have a shorter reader and attach the shorter vessel right there. Here's a double kidney transplant.
oh, on the same side?
Uh-huh. And then a kidney and pancreas and
Would there be no trouble on the same side, Professor? Feeding both kidneys?
they would probably, no. And they showed both of the kidneys up here in place, but if they're diseased or tumors or whatever, they would probably take them out. They're just have them there to show you where they normally would be located.
Huh.
Okay. All right, so let's look at the arrangement structure of the kidney. Get my lights back on. There we go. And I'm going to draw it horizontal just so that I can get most of it above the desk here. Now, I don't know which came first, the kidney or the kidney bean, but it has that shape and similar to lymph node as well. With a hilus and a cortex and a medulla. The medulla stops here. Okay? So the rest of this area here is called the sinus, where you see in between the adipose tissue. So this is actually known as the sinus, and it contains the structures that actually transport urine. So as we'll see starting on Tuesday, when we filter the blood, that first fluid is identified as filtrate, not as urine, because it's components filtered from the blood. Urine does not have amino acids in it. Urine does not have glucose in it unless you're diabetic and glucose is spilled over into the, not been completely reabsorbed. And so we don't actually finish the modification of the filtrate until it leaves these structures here known as the pyramids. Once it empties out of these structures, it enters into tubes or channels known as calyx or calyces for plural. And at that point, we can call it urine. Okay, so I'm going to draw the pyramids in, and we'll come back and talk about what forms these structures. So these are cone-shaped structures that come to a tip. They are formed of straight, vertical oriented structures, tubes and blood vessels, so it gives them this striated appearance. And some of those tubes travel the length of the pyramid. So in this model of a kidney, here's a pyramid that opens up into this calyx. And so coming from the back side of the screen, in the model, you're looking straight on at the tip of one of these pyramids. And all these little circles here are the openings of the tubes. And at that point, as soon as the fluid leaves that tube and enters into this space, it's now urine. We're no longer modifying it. So the tip of the pyramid then is known as the patella. And this entire structure is the renal pyramid, to differentiate it from the medullary pyramid as the brainstem. Again, it's not a flat triangle. Okay, it's cone-shaped three-dimensionally. And then the minor calyx would be the single channel that this would drain into. So that would be a minor calyx. Here would be another minor calyx. So each kidney pyramid drains into a minor calyx. And then two or more minor calyces would join together to form a major calyx. So a minor calyx, that's the singular form. And these would be two major calyces, is plural. So back to our diagram. Here would be a minor calyx. Here's a minor calyx, a single major calyx. Minor calyx, major calyx. Then all the major calyces come together to form a trumpet-like structure known as the pelvis. Okay? So if I were to diagram that up here on the board, just the calyces, the minor calyx, major calyx. And then where it tapers and becomes more narrow in diameter, outside the kidney, we identify it as the ureter. So if I were to carry this on down, this is lying on its side.
So is that going to be past the helix of the ureter?
Right, because it tapers actually outside of the kidney. And then sometimes students have a difficult understanding of the difference between the pelvis and the sinus. The pelvis is that enclosed tube that contains the urine. The cavity around the minor and major calyces that's filled with adipose tissue is the sinus. So let's say you're going to put a pool in your backyard, one of these pre-built fiberglass pools that they just drop in. So you dig up, have a backhoe, dig a big hole. Okay, that would be the sinus. And then you drop in the pre-built swimming pool, that would be the pelvis. And then you backfill in around that. And that's what the adipose tissue is doing here. Okay. Now, all of these structures that are actually draining urine are lined with our urinary epithelium. So remember when we had a respiratory epithelia, that was our pseudostratified columnar epithelium? What would the urinary epithelium be? Transitional. So starting with a minor calyx, which is where we first identify the fluid as urine. So this is the first structure. To contain urine. And then major calyx. Ureter, urinary bladder, and then most of the urethra all are lined with transitional epithelium. Within the kidney itself, the functional structure of the kidney, we'll see simple squamous, simple columnar, and simple cuboidal.
Is that going to be the whole, all three parts of the urethra?
The very last portion in the last half inch or so, inch to inch and a half inch in female, about an inch and a half in male is non-cornified stratified squamous. All right, so that's kind of the transport part of the kidney. And the functional part of the kidney, we have the nephron. So the nephron is what we're going to be discussing next week on how urine is made. But the nephron is, to give you an example, this structure right here. And I'm going to diagram it on the board. Part of the nephron is always and only found in the cortex. Part of the nephron can be found either in the medulla or in the cortex, depending on whether it's a short or a long nephron. All of our nephrons will eventually dump their filtrate into a long tube that looks like this, that travels from the cortex into the medulla, called a collecting duct. The reason I'm stopping at this picture is this is why the medulla looks striated, the pyramids look striated. Because these loops go straight down and straight back up, and the ducts go straight down. The blood vessels that are in here also loop. And so they are straight structures. They go straight down, turn, and go straight back up again. So that's why we have this striated appearance to the medulla. Okay. So I'm going to take one of these nephrons. I'm going to erase the pelvis over here. And actually, I'm going to draw it on the board over there because I found out earlier this morning that I still can't draw it short enough to visualize on the board behind me. So this is we'll do the blood vessels either the end of today or on Tuesday. These blood vessels right here are found in this position. So they are the artery and the vein that are drawn there run along the base of the pyramid. So the outer portion above this vessel is going to be the cortex. Down here would be the medulla. And we'll come back to the blood vessels, but this is known as the arcuate artery. Yeah, because they go to ARC. Yeah, because they arc. Each base of the pyramid is an arc. All right, so the nephron is the functional unit of the kidney. If we go back to our embryonic slide, if you'll bear with me for just a moment. We go back to the development of the mesonephros here. This essentially is our nephron right here. So this relationship between the blood vessel and this cup-like portion of the nephron is where filtration occurs. And the rest of the nephron is where we modify what we filter. Okay? So we dump everything that's small enough that will pass across the filtration membrane into the filtrate. And then we spend the rest of the time going through the nephron, removing or adding, modifying the content. So that's why it's called the functional unit of the kidney, but obviously the blood vessels play an important role. So the cup-like portion is where we find that intimate association with the blood vessels. So I'm going to draw those blood vessels in here now, but I'm not going to bother labeling them. I'll do that later. And again, this is like we talked about the pericardium and the peritoneum and the pleura as a balloon, pushing your fist into a balloon. So if you think of that cup as a balloon, and then the blood vessels getting pushed into it, the inner aspect of that balloon intimately becomes associated with all the capillaries. They get surrounded by that. And so when we draw this inner portion, it's really intimately associated with all the blood vessels like that. Okay? So a very quick way to draw the cup is just to draw it like this and put the blood vessels in there. But there's really a much more intimate association. And that's where filtration is taking place. So this structure, the first structure that contains the filtrate, is, I call it by the older term with someone's name, and that is the Bowman's capsule. It's also known as the glomerular capsule.
Oh, the glomerular.
That's what these capillaries are. barrier line.
What are the black structures in
That's this inner epithelium,
there?
intimately associated with the capillaries.
And this is the first start of the filtration of urine?
This is where filtration occurs. This is where we actually make the filtrate.
So the
As the
inside-
fluid leaves the capillaries and is pushed into the capsule. What, Catherine?
It's not smooth on the inside. It's just kind of-
It's smooth squamous. We'll look at this in great detail through electron microscope and everything on Tuesday. All right, then that filtrate is carried away from the capsule into a tube that is highly coiled. And that coiling increases the length and, of course, the surface area through which that filtrate is traveling. And it's closest to the capsule, so it is known as the proximal convoluted because of all the twisting back and forth tubule. Now, I haven't identified the epithelium, all right? We'll do that on Tuesday, but it starts out simple squamous, then it goes simple cuboidal, and then it becomes simple squamous again, and then simple cuboidal again. So we'll discuss that actual epithelial tissue as we follow the function. Now the proximal convoluted tube will go straight, and then it becomes very narrow. And so this is known as the thick segment. And then where it becomes very narrow is known as the thin segment. Of the nephron loop. So this is the descending. Well, let me just put that one side here, sorry. So the thick and thin segment of the descending limb of the nephron loop. Don't worry, for lab quizzes and exams, I'll allow you to do this. I've been writing all of that out. All right, now the thin segment makes a turn. And it gets thick again. So this would be the thin segment. And the thick segment of the ascending limb. The old name for nephron loop is loop of Henle, and you're welcome to use that if you like. And then the thick segment of the ascending limb travels straight for a period of time, comes here right between the two vessels going into and out of the capsule, and then it also becomes highly convoluted. And that is known as the distal. Convoluted tubule. This portion right here, if it is referred to, is the distal straight tubule. It hasn't started yet becoming convoluted as it goes by the Bowman's capsule. So all of that from Bowman's capsule, from proximal convoluted tubule, nephron loop, and distal tubule is the nephron. So on Tuesday, we're going to look at each of those components and how they function in filtration, secretion, or reabsorption. As you can see on the screen, the distal convoluted tubule ends by attaching to a collecting duct. And more than one distal convoluted tubule will attach to a single collecting duct. The collecting duct will merge with other collecting ducts, and at the papillae will open as a papillary duct into the minor calyx. So what we'll see is something like this. And this would be a papillary duct. And this would be a minor calyx.
Collecting duct goes into the papillary duct?
Yeah, so collecting ducts, several collecting ducts would merge to form the papillary ducts.